Peace be upon you all. Assalamu alaikum and good day to everybody. And Dr. Fuz Rana, thank you for being here to on the Finding Truth podcast.
Oh, I'm honored that you would think of me, and so thanks for having me.
Thank you very much. And for our audience, let's first start by introducing you. How about that?
Sure.
Okay, so Dr. Fuz Rana has earned his Bachelor of Science in Chemistry with highest honors. So watch out, this is somebody who has been studying hard from West Virginia State College, and he has a PhD in chemistry with an emphasis on biochemistry from Ohio University. He pursued postdoctoral studies on cell membranes at the Universities of Virginia and Georgia, and worked as a senior scientist in research and development at Procter & Gamble. So not only a person who has studied well and did his academic research, but also applied it in real life applications. And so, Fuz, you have also published in peer-reviewed scientific journals such as Biochemistry, Applied Spectroscopy, Feb's Letters, Journal of Microbiological Methods, and Journal of Chemical Education. Fuz holds 2 patents and authored a chapter on molecular conversions and intelligent design for The Nature of Nature, and co-wrote a chapter on antimicrobial peptides for biological and synthetic membranes. So plenty to do with membranes. So I hope we have the time to ask you about cell membranes and why they have fascinated you so much. In addition, he has delivered numerous presentations at international scientific meetings. Now, it's one very important question that we are gonna ask, but one very inspirational thing about your story, Fuz, is that in the beginning you were ascribed to evolutionary paradigm, and then you converted to believing in God, to Christianity, after going through your graduate studies. So obviously, your story suggests that studying biology more takes somebody to faith. So that's very interesting. And today, Fuz writes and speaks extensively about evidence for creation, emerging from biochemistry, genetics, human origins, and synthetic biology. And that is what we are going to ask him about. And he also have authored several books, and this includes Humans 2.0, which is a topic that I'm really interested in, transhumanism, and will humans cause their own evolution, let's say, or something. And Thinking About Evolution, which is a very nice book where he addresses, I guess, 25 questions, right? Mm-hmm. About evolution, and a very interesting read. And the next one coming up before the end of this year is Fit for a Purpose, which is maybe we'll touch about what's there, but definitely something that I'm anxious that he issues, and then we might have a separate discussion about it. This is among countless articles, and I really would suggest to everybody watching this to note the ticker, where we also have Fuz's website. It's on reasons.org/team/Fazale-Rana, and there are some fascinating articles there for real food for the thought. So thank you very much for being here, and I'm really anxious to have this started.
Well, thanks for having me again. It's great to meet you and to get to know you, and I'm honored again that you would consider me for your show.
Thank you very much. So let's start by opening. So Fuz, you are a biochemist, okay? And you know, after I post those videos, people who are really advocating evolution would immediately come, why are you having this discussion with whatever? Why not an evolutionary biologist? So I have to explain. So why don't you explain to us what is the occupation and area of study of a biochemist, and why is it relevant to the understanding of evolution versus creation or design?
Sure. Well, I think most people know that chemistry is the study of matter, and what is the structure of matter? How does it undergo transformation? And of course, chemists are dealing at the atomic and the molecular scale as they ask these questions. So biochemistry is simply chemistry applied to living systems. What is the structure of matter that makes up living systems, and how does that matter undergo transformation? But then biochemists are interested in something else, and that is, how does that insight then help understand the function of living systems? And so from my vantage point, biochemistry is really ultimately foundational to biology, because you're looking at biological systems at the most basic fundamental level, and then from that ground level trying to go upward to understand larger scale biological processes. So biochemists are very much life scientists, and today biochemistry is indeed influenced very strongly by the evolutionary paradigm. You have to work in evolutionary theory even as a biochemist, in the climate that we live in today. And so evolutionary theory was always in the backdrop of everything that I learned and worked on as a graduate student, as a postdoc, even to some degree as a researcher in R&D. And in fact, biochemistry really relates very closely to, I think, a central component of the evolutionary paradigm, which is the origin of life question. Now, many people try to separate the origin of life from biological evolution proper, and I'm sympathetic to that point. But on the other hand, the grand claim of the evolutionary paradigm is that everything in biology can be explained by mechanism, and this would include the origin of life, which is really the question about the origin of biochemistry. So I think biochemists do actually have quite a bit to say about evolution because of the way we approach the study of living systems.
Is it also fair to say that since the claim of evolution is that, especially after the discovery of genetics and its establishment in the 60s, that essentially evolution is at its core chemical changes to DNA material, right? It's mutations. Mm-hmm. And the study of chemical changes to biological molecule like DNA is in the core of what biochemists do. Is that correct? Is that a fair statement?
No, that's a very good point, Ahmed, that you made. And and I thank you so much for raising that point, because it is true that biology today is highly molecular, right? And so while this now moves us into the arena of what might be formally called molecular biology, but the line separating biochemistry and molecular biology is really a fairly diffuse line, if you will. And so you're exactly right that because biology has become molecularized, you have to understand macroscopic biological phenomena ultimately in molecular terms, and that would include evolution as well.
And eventually the forms of creatures, or what they call phenotypes, are essentially at the end genetic, or what they call genotypes. And this is at the end an information-bearing molecule which falls under chemistry one way or the other.
Yeah, that's exactly right. Yes. Yeah. And so to ignore one and not the is going to be essentially to your detriment.
So inspired by your story, Fuz, and really, I go into so many discussions where people say, no, you cannot see God in science. It's methodological naturalism, and it's against science to even consider God. And if you tell them something else, they would, just shrug, So in your case, that seems to be very more consistent with what I advocate, which is that science takes people who take it seriously and set aside their prejudice to God. Can you tell us more about how this worked for you?
Yes. Well, as you mentioned in the introduction, as an undergraduate student, I was an agnostic. I didn't know if God existed or not. And honestly, I could have cared less, to be frank. I was just, more interested in other things, particularly really becoming the best student of science that I could to prepare myself to go to graduate school. because I really wanted to be a biochemist, from the very early days of my undergraduate education. And so, I embraced the evolutionary paradigm. In retrospect, it wasn't because I was overwhelmed by the evidence. It was because I admired my professors and respected them, and they were essentially teaching that, the way we understand the origin and the design of life is through evolutionary mechanisms. And so I accepted those ideas more or less uncritically as an undergraduate student. And, if you would have asked me, does God exist at that time? I don't know how I would have answered that question, probably with, again, a level of uncertainty. But to me, I didn't think God was necessary because, I embraced this idea that if evolutionary mechanisms could explain everything, it really doesn't leave any room for God. And so that was my perspective going into graduate school. I had no interest in religious ideas whatsoever. And yet, as I really began to appreciate the elegance and the sophistication of biochemical systems, plus I was being encouraged as a graduate student to think independently, not to be influenced by the ideas of the day, but rather fearlessly follow the data where it leads. And at that point, it really begged the question to me, it prompted the question, how do we explain these incredibly elegant and sophisticated systems that are literally awe-inspiring? And when I looked at, chemical evolution as the explanation, I just simply found that approach to be inadequate. And when you see what appears to be design, and that design can't be explained by mechanism, you really are left with one conclusion, and that is that there must be a mind, there must be a creator that brought everything into existence. And so, that then, of course, leads to very interesting questions that are really theological in nature, and that is, who is this creator, and do I relate to that creator? But for the first time in my life, I was really open to those kinds of questions, but I was absolutely convinced that indeed, a creator exists. Now, I converted to Christianity, but, I oftentimes will make the statement that even if somebody came to me and presented a convincing case that Christianity was false, I would still believe that there was a creator behind everything by virtue of the scientific evidence. I think it's so compelling towards that end. Now, of course, that's not considered today a bona fide scientific explanation because of as you mentioned, the influence of methodological naturalism. But if you relax that influence and just let the data speak for itself, it points you, I think, to the place where you have to conclude that there really is a mind.
So in your experience, I have drawn two conclusions here. Number one, that unfortunately, the current mainstream, if somebody goes to university and he's studying, the mainstream will systemically and systematically take you towards atheism. Is it because they would essentially present you with the case of no need for God, everything else, he doesn't intervene, even the initiation of things didn't need him, and hence, why would you care? So it would at least take you to agnosticism, if not atheism. Is this the unfortunate state of affairs, in your opinion?
I think so. And, I don't know that it's necessarily the overt intent of science faculty to undermine faith of students, but I think it actually does happen because, methodological naturalism is supposed to be neutral on the issue of God, right? Where,
supposed to.
Yeah, right. Whether, the point of view there is that whether you believe in God or not, it doesn't matter. But when you do engage in science, you have to operate functionally as if there is no God, right? You can only pursue mechanistic, materialistic explanations. And sometimes it's called benchtop atheism or provisional atheism. But what it does is, in effect, renders science is thoroughly an atheistic enterprise, right? Because you've excluded the possibility that there is a creator that is responsible for aspects of nature or is intervening in some way to sustain nature or to oversee or to regulate the processes that we see in nature. And so if you are a thinking person and you've embraced methodological naturalism, you really are just steps away from philosophical naturalism or from atheism. So and I know many people who are very devout in their faith, who aren't practicing scientists, who do approach science from the framework of methodological naturalism, but their faith is independent of their work as a scientist, not because of their work of a scientist, I would submit.
All right. So I guess you agree with the first statement. So let's look at the second statement, which would be, if someone does not let himself go with the flow of the mainstream and uses his critical thinking, if the first sip of science will intoxicate you with agnosticism or atheism, just few more sips with some critical thinking will bring you to God. Is this your experience? Is
yes,
this your advice?
It is. I think that's exactly the case. And I'm getting to be old these days. Age is catching up with me. So I came to faith, when I was a graduate student, which was the mid 1980s, and so that was close to 35 years ago now. But at that time there were two books that were very popular among the scientific community. Everybody was reading those books, and they both were written by Francis Crick. One of them is called What Mad Pursuit, where it's a charming autobiographical account of the discovery of the structure of DNA. It's a fascinating read that I highly recommend. And another book that Crick wrote is called Life Itself, which explores the origin of life question. So here's Crick, a Nobel laureate. And some people call him a Nobel laureate's Nobel laureate because of the importance of the work that he did in his lifetime. And he was an outspoken atheist, to be clear. But interestingly enough, he concludes What Mad Pursuit with this incredible statement that biologists must constantly keep in mind that what they see was not designed, but rather evolved. And then he talks about this idea that the origin of life appears at the moment to be a miracle. So many of the things that need to be in place to get it started. So Crick actually came to the same conclusion I did as a graduate student. Not that I'm putting myself on the level of Francis Crick, but Crick as an atheist refused to follow the evidence where it leads, right? Whereas for me, I would humbly suggest that I did follow the evidence where it leads. And so I don't think the question of design is being disputed. It really is who's responsible for that design, or what is responsible for the design. And I don't think the question of challenges to the origin of life are being disputed. The question is, what do you do with those challenges, right? And so I don't know why people refuse to go down the theological paths when that's where the data is leading, but people seem to be very uncomfortable doing that. But if you're willing to do it, I think you find ample evidence for God.
That's great. So that's been very insightful. The matter that somebody in the knowledge of Francis Crick at the end says it's a miracle, but refuses to acknowledge God is also disappointing, unfortunately. But let's now, if it's not God, what is it? What is this Darwinian evolution, or recently called modern synthesis or extended modern synthesis thing, proposing as a replacement to God? So let's first ask the first question. What is the claim of Darwinian evolution? What is sitting in the place of God there?
Yeah. And to me, and I don't want to take too much time with this, but it's really instructive to look at what was happening in biology prior to Darwin's publication of Origin of Species. And for the most part, biology was teleological. People saw design and purpose in biology. Now, there were debates about whether it was function or form that was the most important feature of biological systems. And so there wasn't consensus as to exactly how to interpret that design, but people did indeed see a teleology to biology. Darwin comes along and he basically argues that evolutionary processes mechanisms, natural selection, can actually explain not only the origin of species, but Darwin argued, if extended over long enough periods of time, could explain the totality of biology, including the exquisite adaptation of organisms to their environment and the incredible design that we see. So what Darwin did is he replaced the mind of a creator with mechanism of natural selection, and later added sexual selection to that suite of mechanisms that could explain the diversity of life. This is called the Darwinian Revolution, in part because he essentially he turned biology upside down. He stripped biology of all teleology. And so to me, when I think about the evolutionary paradigm, of course it is, descent with modification, where there is a last universal common ancestor that through evolutionary processes produces this vast evolutionary tree of life through this descent with modification. But also a very important aspect of modern day biology and of the evolutionary paradigm is anti-teleology, is that to suggest there's purpose, even in the evolutionary process, to say that there's purpose even in evolution is considered to be completely forbidden.
It's a crime.
Yeah, right. So in effect, when somebody embraces theistic evolution, if you're going to adhere to the scientific mainstream, you have to adopt an anti-teleological approach, which is incompatible with theism. Now, there's some very bold thinkers that I admire who are proposing an idea known as structuralism, where they say that actually there is deep-seated purpose embedded within the evolutionary process that arises out of the constraints of the laws of nature that forces evolution to predetermined, prescribed endpoints. Now, that is a highly teleological view of evolution that, again, is frowned upon by the mainstream. But at least if somebody is approaching evolution from that perspective, I'm sympathetic, because they are still seeing a role for a creator ultimately in nature. Whether that creator is working directly or indirectly, the creator still involved, and there is intent and purpose. And so, not to belabor this point, but I think people really need to appreciate how strongly the evolutionary paradigm is opposed to teleology in any shape or form. And it's very hard.
Teleology is design, actual design, not apparent one. Yeah,
that's right. Yeah. So biologists will play around with design terminology and use this concept of teleonomy, which basically is a $25 term that makes it seem, that you're highly academic, and sophisticated when you use design terminology, but it's just a card, a get out of jail free card that allows them to use design terminology, but really not mean design. But it's interesting that even in an anti-teleological context, you cannot help but to talk about the design of these systems and treat the systems as if they really were designed. It's unavoidable, and that to me suggests that maybe they really have been designed for a purpose.
Yeah, and to sum this up, it's the neo-Darwinian or the Darwinian claim is that everything goes back to a single point, and what has driven it out is just random events that are selected through this blind, unguided natural selection mechanism. So it is essentially mechanism replacing mind and chance replacing intent. Is that, and
yes.
that through those you can explain everything about life. That's the claim.
Yes, very well said.
And now, obviously, when anybody looks at life, and we recognize a bacterium as living and a cat and a human as living, and there's so much differences. And as an engineer myself, I don't really understand how somebody in his right state of mind will accept this. But this is just me. Maybe I'm just the lay person here. As a specialist in this field, as a PhD in this field, do you think that this Darwinian mechanism, natural selection acting upon random mutations, can explain all this novelty that we see from single-celled organisms, unicellular organisms, to the human being? Is this possible in your opinion?
The short answer is no. And I think evolutionary mechanisms, are very powerful at manipulating an existing design, in fine-tuning it, in adjusting that design. But I really see very little evidence that those mechanisms of natural selection, sexual selection, and genetic drift can actually create categorically new types of designs. And so I'm highly skeptical of that. And Darwin is to be credited with discovering natural selection and a mechanism by which species can adapt to their environment, where one species can give rise to a closely related sister species. So this isn't in question. And this aspect of evolutionary theory is very useful to explain, I think, certain features of biology. It helps us to explain, the origin of certain types of infectious disease agents, things like that. So it's a valuable idea, to be certain, but I see very little evidence that this mechanism can somehow be extrapolated to explain categorically new designs. And this is, I think, increasingly being acknowledged by specialists that are working in evolutionary theory. This is the motivation for the extended evolutionary synthesis, is to recognize that these mechanisms don't seem to be able to account for things like the origin of new body plans.
This is our next question. So,
yes. Okay.
I have been struck by that event in, I think it was 2016, the Royal Society, where it started coming to the mainstream, the extended modern evolutionary synthesis kind of nomenclature. And the thing there, for those who didn't hear about it, is that the top evolutionary biologists just came to the table, put it in the open, that the Darwinian mechanism is not enough, and they proposed those other mechanisms. And they just called the whole collection this extended modern evolutionary synthesis, as a replacement to the traditional neo-Darwinian modern synthesis. But in your opinion, is this really covering the gap? Is this the deal? Self-organization, neutral theory, the other stuff, is that enough? Are we really at rest now?
Oh, I don't think so. I think what is, I think, significant here is that because of the extended evolutionary synthesis, first of all, the critiques that have been leveled against the evolutionary paradigm by people that hold to a creation perspective or intelligent design perspective are now being recognized, at least indirectly, as having legitimacy. Because when experts in the field are saying the very same things that again, creationists and ID people have been saying for several decades, it gives legitimacy to, again, I think, critiques of the evolutionary paradigm. But really the whole point of the extended evolutionary synthesis is that we've got more work to do, that these are possible new mechanisms that we need to incorporate into evolutionary theory, but nobody has shown that these are going to somehow solve the problem. It's just that these are now new attempts at trying to bring resolution to it. And part of this synthesis, extended evolutionary synthesis, is work in evo-devo, right, evolutionary developmental biology, where people are recognizing that if you're going to explain the origin of body plans or the origin of new designs from an existing biological design, you've got to exert evolutionary influence at the developmental level. The problem is that in order to alter designs fundamentally, you have to exert those genetic changes very early in the developmental process. And we know from laboratory work on, let's say, fruit flies and other organisms, that you can indeed, through very small changes in developmental genes, have large-scale effects on the design of the organism. But invariably, those changes are catastrophic. You create monstrosities if the organisms actually even survive. And so how do you affect changes at that level that produce these new, not only body plans, but new body plans that allow that organism to somehow have an advantage in the environment? That to me is far from being established. In fact, I think the work in Evo Devo is suggesting the opposite. And what's interesting is that the further you go into development, that's where you are able to introduce genetic changes that actually are not catastrophic, that may, again, fine-tune existing designs. But now that's pushing you from macroevolution into microevolution and speciation. So what you see in the work in Evo Devo is actually, again, an affirmation, I believe, that microevolution and speciation are legitimate aspects of evolutionary theory, but to claim that you can create novelty, even with the extended synthesis at this point, is far from established.
And I think you're referring to developmental gene regulatory networks when you're talking about the why Evo Devo is taking us more towards design, because the wiring of those things are really unbelievable. Am I correct to assume that this is what you're referring to?
Yeah. Yes, exactly. Things like the Hox genes, and the thing is that it's interesting to me, because I oftentimes will see biologists say, well, we understand the process of development now at a genetic level, and what they've really discovered with these Hox genes are on-off switches. They still don't understand the genetic regulatory networks, which are probably a network of networks, in fact, that have to be triggered to create, a particular aspect of the organism's phenotype. Nobody understands that. They just understand the on-off switch. So it's like me saying, well, I naively know how my television works because I know how to turn it on and off. Well, yes,
the buttons of the remote, but
but
that's not enough at all.
right. And so but it's not fundamentally explained. And just because I can take maybe that on-off switch from my TV and put it on, let's say, a CD player, now I'm old school, giving away my age. You might be able to take that mechanism and move it from one device to the other and get that device to turn on and off, which is what you see a lot happening with work in, evolutionary developmental biology, where people are moving these genes from organism to organism. But it still doesn't mean that you understand, again, how a CD player works relative to a TV, in the differences, nor does it explain how a TV could evolve into a CD player. So this is a very simplistic analogy, but in effect, I think it helps to give people a sense for what has been accomplished, which is remarkable on one hand by the scientific community, and yet what is left to be understood, which is a huge chasm in understanding. That if it's not understood, there's no way you could legitimately say that we have explained the evolution of novelty.
Great, and it reminds me from my field with the fact that a TV and a CD player are, they're essentially the same components, transistors, capacitors, coils, resistors. They are put on the same boards, and essentially the laptop and the mobile phone and the TV and the CD player, the fact that they are all the same components doesn't, and they can all be hierarchically categorized into electronic equipment with various variations and classifications, doesn't mean that they evolved into each other, neither that they have evolved from a common electronic ancestor at all. Right. It's just us electronic engineers doing these, little pieces of design and bigger pieces of design, and using them and shuffling them to produce different equipment that share common features, but are not at all evolved from each other. So I think this is a great similarity between electronics and biology too. So it struck me to say it.
Well, Ahmed, you're raising a really, really important point, because the primary evidence that I see people cite in favor of an evolutionary history of life are the shared biological features. And what you're pointing out is that you could understand those shared features as reflecting shared design as opposed to shared evolutionary history. And in fact, it's interesting to me that prior to Darwin, you had biologists like Sir Richard Owen, who was viewing biology exactly in that way, where he argued that there was these archetypical designs that existed in the mind of the first cause that were then functionally manifested in the created order, and what he marveled at is that these archetypes were so robust that you could actually manipulate them to create a wide range of functional systems. And he argued that this is the quintessential evidence for design. It's not the form or the function, but it's the fact that the form supports the function, but supports a diversity of function. And so what you see is these exquisite engineering principles that are being played out in biology. And, I've had skeptics say to me, well, if you're talking about an infinite creator, why wouldn't that creator just create an infinite number of designs, right? And to me, the point I make is that if you think about this for a minute, if it wasn't for these shared designs, it'd be very difficult for biology to even exist as a scientific discipline. It's because of these shared designs that we can study, a nematode, or we can study the yeast cell or the bacteria E. coli and extract principles about how living systems work that are universally or near universally applicable. And so biology is not only designed to be highly efficient, but it's designed to be discoverable, right?
Yeah.
And so that to me suggests that this creator wanted us to understand the world around us, part of, I would say, the creator's providence, but also it reveals the creator to us. And so, it's the same idea. Think about, your discipline of, electrical engineering, right? If it wasn't for the modularity of devices, if every electronic device was unique, engineers would never be able to truly function, right?
Absolutely.
Right.
And that's why we have what everybody knows as ICs, integrated circuits. You go to the electronics shop and you buy this IC, and this IC is a common component across hundreds of devices. But also, it's very important in something else that you might as well respond to here, because if it was not for ICs, for example, recycling would be very difficult. If a TV is dead, the only place it can go to is the recycle bin. Now, of course, in our economic environment, maybe it still goes to the recycle bin. But if we have scarcity of resources, some very smart people will be picking out ICs from those printed circuit boards and using them for other devices. And in biology, just tell me your opinion about this. If every creature was its own unique thing with its own unique biochemistry or chemistry, could we, at the end of the day, eat animals and plants eat, have bacteria cooperating with them, and then bacteria causing everything to decompose and becomes part of their bodies? All this cycle of life, I think the whole planet Earth would have been filled up with biochemical trash in no time. Isn't that also something?
Yeah, that's a really provocative point, and I would agree with it. I think that that's a really, really good point about just the necessity of this modularity in order for symbiotic relationships to exist, for example, for ecosystems to exist, of course, and again, for the efficient cycling of nutrients. You might argue that maybe if every organism was unique, or at least every species was unique, you could still break everything down to the fundamental building blocks, but it would be incredibly energy intensive and inefficient to go from the bare building blocks to build separate classes or categories of biomolecules, and probably the diversity of life would be limited because for that reason as well. So it's an excellent point, that modularity has broad implications.
And maybe I would take from what you have said that the common design is a gift from God to us to infer the common designer, isn't it?
Yes.
If everything looked different, maybe polytheism would have been, believing in 30 million gods or something. Each one created its own creature. might have been a better proposition than monotheism. But I think this is a real strong case for monotheism. It's the same creator who has created the physical laws that cause chemistry to work and make biochemistry possible and make biology possible. And guess what? It's ubiquitous. It's everything using the same thing. It has to be one. Isn't this a compelling case?
Yes, because that was, one of David Hume's criticism against the design argument. How do you know it's not a bunch of designers instead of a single designer, right? And I think your point is, really well made, and that is, this is highlighting the fact that there is a single designer who does have the power to create an infinite array of organisms, but chose, I think, to limit how he went about creating in such a way to reveal himself to us, but also, again, to provide for our needs.
That's great. So we've talked about design. Before we just jump into your creation model, we run out of time, but this is really interesting. So I see you in your writings, you frequently refer to the anthropic principle. And I want to ask you, what is the anthropic principle and why do you think it relates to evolution versus creation discussion?
Yeah, well, when people think of the anthropic principle, they immediately think of astronomy and physics, right? And it's this idea that the fundamental constants and the parameters and the characteristics of the universe that have numerical values that define the universe have to assume exacting values in order for life to be possible. And if that any one of these quantities deviated, in some cases almost infinitesimally, from the value that they hold, the universe would be essentially incompatible with life. There's no way that life could exist in the universe. And so the line of reasoning there is that the universe is just right, that the constants of the universe are just right to make life possible. And the way you show that is by showing that there's not alternative values. And in fact, in the book that I hope will soon be released, Fit for a Purpose, I make the argument that this idea of the anthropic principle actually extends into biochemistry. That is, the molecules that we see that make up living systems have a just right set of properties that are ideally suited for life to exist, and that we can show that there's not alternative molecular systems that could substitute for the systems that we see in the cell. And so they seem to be unique. And I show in the book that these systems seem to be fundamentally prescribed by the very laws of nature that seem to naturally produce these systems. And so that to me is a bit eerie to think that, that the laws of nature are producing these just right systems that are unique and have the capacity to support life. So it's an anthropic, argument, the anthropic principle being applied to biochemistry. But that to me, is interesting, because if indeed there is a creator that designed everything to be just right, why would that just simply be limited to the laws of physics? Wouldn't you expect to see it in chemistry? Wouldn't you expect to see it in biochemistry, and maybe even in biology itself? And so, it's an idea that I don't think many, people that argue for on the basis of science for a creator have explored. A few people have dabbled in it. So I'm hoping that this idea will actually, again, further emphasize the design that we see, in biochemistry.
So let me rephrase my understanding. What you're saying is that it's not only the fine-tuning of the constant of physics that we can see, but we can also see that the fine-tuning of the very physics almost compels chemistry to lead to biochemistry, to lead to biology in a way. To me, it means few things. Number one, that the fine-tuning extends all the way to human beings. And number two, that there is no, God of the stars and God of the moon and God of animals and God of humans. It's the same one, and you can see his signature
right
from Big Bang to human being. Right? And it couldn't have been two. it couldn't have been two cooperating together or something, because it's just exactly, he's doing it exactly the right way, to the extent that he just needs to ordain it, that life comes to being, but the components and the chemical interactions and the chemical properties are engineered into the physics. Is that it?
That's exactly right. Yeah, and the way that you show fine tuning in biochemistry isn't through like these theoretical analyses like you see in physics, where you're varying the values of the different constants and you're looking at the effects that would have on the universe, but rather you actually are doing it experimentally, where you can try different molecular designs and see, do these
which we cannot do in physics, because
right.
we cannot change the gravitational constant in physics.
Right. Yes. Yeah. So this is so it's qualitative versus the quantitation you see in cosmology, but it's, I would argue, is more robust perhaps because it's experimentally determined, and the scientific literature is just filled full of the work that people have done to unwittingly that supports this idea, and so what I really did in the book is just kind of pulled all of this work together, into a coherent framework that I think really shows that there's something very suspicious going on in biochemistry. Either we got really, really lucky, or there's really a mind behind everything. And
this is the Fit to Purpose book, right? The
yes.
upcoming.
Yes.
Yeah. You made it even more exciting to wait for it to come out. Yeah. So I want to jump into those examples, but I'll just really run out of time here, and this is unfair. But let's go into creation. And first, let just give us in a nutshell. I've seen you referring to RTB being reasons to believe, I think, yes. A creation model. So this is a creation model. It's not theistic evolution, that's for sure, because as you have rightfully mentioned, theistic evolution to me seems contradictory, to say that a random event causes by being guided by God, while the evolutionary theory itself says there's no guidance. So if you accept evolution, you cannot. These are two mutually exclusive things. How does RTB creation model solve it, and what is it?
Yeah, well, our creation model is essentially we would call ourselves, old Earth creationists. So we accept the antiquity of the Earth, the antiquity of life on Earth, the antiquity of the universe, but that we are suspicious of at least aspects of the evolutionary paradigm. So in that sense, we're creationists, but yet we hold to an old Earth perspective, which is distinct from a number of our Christian brothers and sisters who argue that Scripture is teaching that the Earth is only 6,000 years old. So we're creationists, but we reject that particular version of creationism, if you will. And we would argue that in Genesis 1, days are a period of time. And so we would say that that's the way you read Genesis 1, is that it's referring to epochs, not 24 hours. So that's
you're aware that this is the mainstream also interpretation of the 6 days in Islam.
Yes.
Muslims typically don't have the YEC issue. It's typically okay. It's 4 billion years. It's okay. We don't mind.
Yeah. Well, it's interesting because I had a chance to work closely with some Islamic creationists in Turkey, and yeah, that was the young Earth, old Earth thing was not even.
It was not an issue.
It was to me. It was refreshing. It was refreshing not to have to go down that path. But yes, I'm familiar with that, and it's a delight to learn that, by the way. But our approach at Reasons to Believe is to try to take what we understand to be what the creation accounts are teaching in the Old and the New Testaments, and then from that to convert that insight into a scientific model that actually makes predictions about what would be discovered if there's a creator, what would be discovered if these creation accounts are valid. And what we're not trying to produce an alternative scientific model to Darwinism, but rather what we're trying to do is create an approach that invites scientifically minded people to test our ideas. scientists think in terms of, how do I test the idea? How do I falsify this? How do I affirm that this is true? What are your predictions? And so it's a way to speak the language of scientifically minded people and invite them, into, a conversation about whether or not there really could indeed be a creator. So that's really the intent, but we are not a theistic evolutionist at all. While we do see an antiquity to life on Earth, and again, we're comfortable with some aspects of evolutionary theory, at the end of the day, we see really ultimately God intervening in direct personal ways to bring about his creative purposes throughout the history of life on Earth. We see clear fingerprints for the creator in nature.
Let's then ask about those clear fingerprints, but I'm not going to ask you an open question now because we have limited time. So I'm going to jump immediately into one of the very interesting articles that I have read from you, which is about Krebs cycle. What is Krebs cycle in a nutshell, and what is its significance to this creation?
Yeah, well, the Krebs cycle is, perhaps one of the most important metabolic pathways in the cell. It really lies at the very hub of what's called intermediary metabolism. So this is the metabolism that essentially brokers in small molecules, usually pathways that are building the building block materials for proteins and DNA and RNA and cell membranes, or is breaking down materials either to eliminate them as waste or to harvest energy for the cell to use. And so but the core pathway that is like the hub, which gives rise to all these other pathways, is the Krebs cycle. And many people think that this, from an evolutionary perspective, very well may have been the very first metabolic route that would have originated. And what I do in the article is I talk about it's how remarkable it is that the molecules that make up the Krebs cycle seem to have an unusual set of properties that not only make them ideally suited to serve in their role in metabolism. This is the idea of the anthropic principle again, but also have this unusual set of properties that makes it appear as if they are going to very readily form under the conditions of the early Earth. And in fact, people have shown that you can start with molecules like glyoxylate or pyruvate and actually produce the Krebs cycle intermediates very easily. And so it's really eerie to think that here you have these molecules that have the just right set of properties that make them ideally suited to serve as the centerpiece of metabolism, that there's not alternatives to the Krebs cycle that anybody can conceive of. And people have tried, by the way, and yet that the laws of nature seem to be rigged in such a way that these materials are already available on the early Earth. And so this is, again, where you start seeing these anthropic coincidences where you have the just right materials that are prescribed by the laws of nature. Because, the laws of nature could constrain, the chemistry on the early Earth in such a way that these materials would never form, right? But it doesn't. It's constrained in such a way that these materials form, and they happen to be, again, the just right materials that you need, to sustain at least the core of intermediary metabolism. That to me is really very eerie, and suggests design at a very deep-seated level within the nature of the universe itself.
Actually, when I read your article, the following idea struck me. It's like, to me, what is giving hope to the origins of life researchers is that when they do certain things, they find certain results. And because they are driven by methodological naturalism with philosophical naturalism as their backdrop, it gives them self-affirmation. It's a positive reinforcement cycle that, hey, look, those components formed in, under 70 degrees, and this looks like good for a vent or something. And what you're saying to me is that what they don't realize is that those laws are there so that when God ordains life, life will not struggle to access its basic elements.
Yes.
And then what it predicts to me is that origins of life research will over and over again do experiments that will superficially give the impression that life is a natural consequence of physics, but always stop at the point where somebody will need to draw the blueprint of the resistor and the capacitor and the transistor. Otherwise, they will never come together into a circuit, and definitely not into a laptop.
Yeah.
Is that a proper insight from what you've?
Yes, that's very insightful, and and it's important to note that even though these components readily form, or appears to be able to readily form under the conditions of the early Earth, which again is it's like things are conspiring, for these materials to form. They don't naturally self-organize into an autocatalytic network, which is what would need to happen, right? So nobody has shown that this happens, but rather that these materials are present. The analogy that I use isn't maybe quite as sophisticated as yours. I really like your analogy, but it's imagine that I'm going to build a puzzle, one of those jigsaw puzzles. And so I have a picture on cardboard, and I very carefully cut out the pieces of the puzzle. So the pieces of the puzzle are designed now to fit together, right? But when you just put them in the box randomly and shake them up, you're never going to get those pieces to actually fit together to form the puzzle. You have to have somebody outside the puzzle, an intelligent agent, deliberately assembling them. But you can just never assemble random pieces of cardboard into the puzzle. Somebody has to design the puzzle pieces so that they will fit together. And so that's how I think about it. Is we've shown that the universe is capable of producing the puzzle pieces that we need. And that in and of itself, bespeaks of intent and purpose, but you still need that creator to come along and I think, intervene in a way to how the creator does that exactly. I don't know, but it's very clear that you need an intelligent agent to intervene to fit those pieces of the puzzle together. I think your analogy is maybe more sophisticated, and probably the complexity of what you're describing is more in tune to what's happening in biochemistry, but yeah, that's my simplistic analogy.
It's not simplistic at all, because essentially even the guy who's putting together the puzzle, let's say the puzzle is drawing, an elephant or something, he has to be somebody who knows what an elephant is, not a blind person who has never seen an elephant. So he must know what an elephant is, and he cannot be blind. So it strikes me that how can somebody think of random mutation, which is completely somebody shaking the puzzle pieces, and natural selection, which is blind? So something that doesn't know what an elephant is and something that is blind will essentially put together the puzzle. Yeah. And his only evidence is that the puzzle pieces are there. This is not the thing.
Yes. Yeah. Well, and what you're talking about here is really foresight. I mean, when you think about the Krebs cycle, it's not just simply that we got lucky and the right, molecules are present on the Earth that you need for the Krebs cycle, which, by the way, has the just right set of properties. But when you think about what the Krebs cycle does and how it integrates all of intermediary metabolism, there was a foresight to, that this is the type of system that you need, and that in order for, intermediary metabolism to exist. So the design that you see in the Krebs cycle is anticipating the totality of biochemistry. That to me is remarkable, is that there's a foresight that's embedded in this design that I think is completely overlooked by evolutionary biologists.
I wouldn't take it as an innocent overlooking anyway, but okay. So the next thing I want to ask you about, first, this was amazing talking about the Krebs cycle. We didn't do it fairness because I was really fascinated reading your article about it. So maybe this needs a complete dialogue on its own virtue. But the second thing, which really caught my attention, because I made this episode about kinesins and dyneins and motor proteins, the transport system in eukaryotes. And I know they are not there in prokaryotes. And then I run into this, the diffusiophoresis. The name itself just needs explanation. And I'm fascinated about it. So just tell us about it, and how does this thing make small organisms work? And then I'll just give you a hint about why I find it so interesting to compare them to kinesins. So
Sure.
yeah.
Well, I mean, I think everybody is familiar with the idea of diffusion, right? Is that, at a molecular scale, you have molecules moving from a level of high concentration to a region of low concentration. And so the molecules are moving along their own concentration gradient. And I believe in the 1940s or 1950s, physicists discovered this phenomena of diffusiophoresis, where molecules will move in response to another molecule's gradient. And so molecule A may have a gradient that goes from A to A prime, but that as the molecules move along that gradient, that molecules that belong to to A, it actually causes molecules that are of a different molecular species, let's say B, to actually move as well. And so molecule B is moving in response to the gradient of molecule A, and this is called diffusiophoresis. And it turns out that
and in this case, molecule B will not have its own motivation from diffusion to move. It's moving because it's like A becomes the carrier for B.
Yes. And it's a very complex surface phenomena that's going on that really is counterintuitive. If for somebody that understands physics, it really is a counterintuitive behavior, but yet it's been well established. And so just recently, a team of researchers showed that diffusiophoresis may actually be the mechanism by which molecules in simple prokaryotic cells can actually move around in the cell. And so they set up, we won't go into the details, they set up this very elegant experimental design showing that you actually do see this phenomena of diffusiophoresis involving what is known as the min D, C, and E molecules that are involved in essentially the process of cell division in prokaryotic cells, where the movement of these molecules back and forth from pole to pole in the cell helps to position what's known as the FTSZ ring that contracts and constricts the cell into two daughter cells. And so they showed that this mechanism of diffusiophoresis seems to be at play, in prokaryotic cells. And so that is, the molecular gradient of one biomolecule can actually cause the movement of another biomolecule. And so they think that this may very well be a universal phenomena, at least in prokaryotic cells, and actually now allows for transport mechanisms to exist that are solely physical in nature prior to the emergence of these more complex, elaborate, systems that you're talking about that are involved in movement of cellular cargo. And by the way, the molecular movement in the cell is absolutely indispensable. It's similar to an economy, right? That it's one thing to produce, something at a factory, but if you don't have a system to move those materials that you produce at the factory to the places where they're going to be used, then everything breaks down. And this is true in the cell. It's one thing to make the molecules, but the molecules have to go to the right place in the cell, even in simple prokaryotic cells, for life to be possible. And so the fact that these researchers discovered this phenomena, again, is fascinating, but it's remarkable. And this is where the anthropic principle comes into play again. Is like, here's this highly unusual phenomena that is counterintuitive, that is fundamentally dictated by the laws of nature, but is the just right mechanism that you would need to support the movement of cellular cargo in simplistic systems. And that makes life possible. So again, there's this these remarkable coincidences that exist that are stacked up on top of each other that suggest design.
That suggests that they are not coincidences,
Yes,
essentially.
yes.
Actually, what I understood from your article, which is very nicely written, is that if not for diffusiophoresis to intervene, cells wouldn't have divided nicely from the middle. I mean, very simple prokaryotic cells, like bacteria, they wouldn't have nicely divided from the middle, because this constriction ring might as well have formed just right at the tip, essentially obliterating the cell rather than causing it to divide, destroying it, chopping it in pieces, essentially, rather than making a proper cell division. And what struck me too is that the material that is pushed in the center is also a material that can recruit further enzymes to reconstruct the cell membrane at this place. And I'm thinking, so if not for this physical property, life would have just died, would have just stayed there dead in its tracks, because even if the first protocell forms, it would have gone nowhere. It would just die and not divide.
Yeah.
And the reason this stunning insight about kinesins and motor proteins is that motor proteins not only move cargo, but they have this unbelievable role in chromatid division. And all the microtubules and then the dyneins are walking, and there's this crazy cartoon rendering that shows them. I did an episode about it. And the bottom line is, without the kinesins and the dyneins doing this crazy magic and the microtubules and all the integration with the ATP-ADP cycle, cells, eukaryotic cells, animal, cannot divide too. And the eukaryote, according to your definition, is too big. Diffusiophoresis cannot do the trick. And we do not have the kinesins and the dyneins and in prokaryotes. So there is no explanation from evolutionary biology, because there is no lineage to these things.
Yes.
So it is as if, to me, the mechanism is needed for prokaryotes. You have the diffusiophoresis, which is essentially a physical property of fluids, but it's not enough for eukaryotes. And then we find those nanobots walking, and those tracks like railways extending to them. And as a biochemist, is there a biochemical explanation that kinesins and dyneins and those motor proteins would do the heavy weightlifting in eukaryotes because they're needed? Can evolutionary theory explain this kind of stunning novelty, in your opinion? Somebody threw at me, well, there is this pro-A and pro-B, whatever proteins that might be precursors to some of the clusters of kinesins.
Right.
Is this something really? Is this really explanations?
Not really, because there are some people that argue that, as you're saying, that there might be these quote unquote evolutionary precursors to microtubules and that are found in some prokaryotic cells. Right, but what you're describing is just because from a design perspective, you could see those similarities of designs as, again, being a modular design, right, that is just being deployed in very different ways. But to go from a prokaryotic cell to a eukaryotic cell with these kind of sophisticated mechanisms, there's not a clear transition of how that would actually happen. And the more that we're learning about like the basic core systems of eukaryotic cells, the more that we're discovering, that the last universal eukaryotic ancestor is incredibly complex, that seems to already have a lot of these systems that are unique to eukaryotic cells that define their complexity. So there's the sudden appearance of eukaryotic cells. It's called the eukaryotic Big Bang, where you just don't seem to have these intermediary states that you see or can even conceive of. It's just that you go from prokaryotic cells to eukaryotic cells. And so people might be able to point to, quote unquote, some clues as to how this happened, but just identifying clues doesn't give you a full-orbed explanation. It's a huge mystery in evolutionary biology, how you would go from a prokaryotic to a eukaryotic cell. And the level of sophistication, as you're highlighting, is stark. The difference in sophistication is stark when you compare those two systems. For movement of cellular cargo, as an example.
I will combine question 3 and question 4 and ask you this. It just came to me now about the anthropic principle, because in Krebs cycle you're looking at part of the cycle happening inside mitochondria, and then
Yes.
the chemical components coming from the cytosol from outside, whatever has converted glucose to pyruvate or whatever has happened before, coming in, and parts of the enzymes attached to the inner membrane, and then ADP and ATP doing all those stunts.
Yes.
And then I find that kinesins, when they're walking, they're using an ATP-ADP hydrolysis cycle to walk. So you have this ATP-ADP thing. It's in the phase 1 and phase 2 metabolic cycle of glucose outside of the mitochondria. They're used again inside the mitochondrion, and they're used to make a kinesin work. And is this magic or what? Why is this chemical reaction using so many things? To the extent it's breaking up molecules, it's making something walk, it's making producing energy, it's doing all these things. Is this in the same line of thought? How things are, amazingly reused and reused and reused in completely unbelievable applications?
Yeah.
Would this be ingenuity of design, the genius of design in biology? Is this part of the genius of design in biology, in your opinion?
It is, and this is something that ended up not making it into the book Fit for a Purpose, which was essentially the rationale for why ATP is used, but it
so it's not only me, huh? Good.
Right, but there is what you're hitting upon here is a really elegant design principle that really is the basis for a highly efficient economy. Think about it. If it wasn't for money, right, then when we're trying to engage in economic activity, it's all bartering. Where if I want oranges and I've got shovels, I've got to find somebody who's going to trade shovels for oranges, and if I can't, then I got to find someone that'll trade shovels for another commodity that the person who has the oranges wants. That's an incredibly cumbersome system. But if you have money, right, if you have a currency, then all I have to do is exchange my shovels for currency, and I can go buy whatever I want, because the person who has what I want will accept that currency. That's an incredibly elegant way to run an economy. This is exactly what you see going on inside the cell, is that ATP is sometimes called the energy currency of the cell, is that ATP is produced by all these energy generating pathways. And by the way, the enzyme that produces ATP from ADP, the ATP synthase, is this incredibly elegant molecular motor that just screams of design in and of itself. But then the ATP can be used to drive all these different diverse processes inside the cell, producing ADP and AMP as breakdown products that then can be readily recycled back into ATP. So it's an incredibly elegant, highly efficient system that is exactly how somebody would design an economy, and it's essentially makes sense that cellular economy would be designed in that way as well. And so and it turns out that ATP has this unusual set of properties that I don't have time to get into, that makes it ideally suited, as an energy system, from the phosphoanhydride linkages that join the three phosphate moieties together. That's again a highly unusual bond that is ideally suited for to store and then to release energy sufficient for the cell to use. And then on top of it, the components of ATP are those components that would be the least expensive to produce from a bioenergetic standpoint. And so it in and of itself is an efficient molecule, where its production, the production itself uses minimal amount of energy inside the cell. So anyway, again, everything that you look at when you look at biochemistry is, highly, efficient. It's highly optimized. There's an ingenuity, a rationale for why it's this way. And it turns out these molecules seem to be just right, for life to be possible. And it just has this coherent fit, suggesting design.
Great. I wanted to ask you about cell membranes, but I think this is a whole huge topic. So I will jump to from seeing creation so obvious to what to me was some today would consider to be a good argument for evolution. So essentially, this thing, when we were looking at animal forms, it was, look how those fossils. And now it's like genetic fossils are the name of the game, or it's the new trend. So under that, some or many actually are advocating that ERVs, or endogenous retroviruses, are good evidence for evolution. So how about taking that on, and maybe like in the 10 minutes and then closing in the remaining? Yeah. So first, can you explain in a nutshell what's junk DNA, what ERV has to do about it? What are ERVs anyway? And why is it important in the human ancestry, or the great ape claim of human ancestry dialogue?
Yeah. Well, there's this view that still largely persists, though the view is very rapidly eroding, that a significant portion of the genome of organisms, including the human genome, consists of non-functional DNA. And there's different classes of non-functional DNA that presumably are introduced into the human genome or the genomes of other organisms through a variety of different biochemical mechanisms. And so the argument is that these non-functional pieces of DNA, again, comprise, in the case of the human genome, maybe close to 98% of the human genome. And so why would a creator produce a genome that consists of non-functional, so-called junk DNA that has
98%. Somebody is bold enough to say that 98% of us is junk.
Yes, that's right. Yes. And in fact, superficially, it looked like that was indeed the case. But yet, what we have discovered is that virtually every class of junk DNA has, function. In fact, there's a growing consensus that a vast proportion of genomes are actually functional in ways that we could never have even imagined. But prior to this emerging insight, people would argue that not only do you see junk DNA, but the different types of junk DNA elements seem to be shared by humans and, let's say, the great apes and other organisms, and they show up in corresponding locations. And so it looks as if these junk DNA sequences would have originated in a common ancestor, and then would have had been retained in the genome, as these different this ancestor gave rise to different evolutionary lineages, and that you could even build evolutionary trees, presumably, with these different junk DNA sequences. But again, if you can show that these sequences are functional, you now have an explanation for why these sequences are in the genome from a design or a creation framework.
So their argument is that since they don't have function, but they are available in what morphologically looks to be an evolutionary path, then the only explanation is that humans are coming from great apes, because otherwise, if this is something that has no use, why is it there? It has to be an artifact of inheritance. This is more or less what they're saying, yeah?
Yes, that's exactly right. And hence, that's why they're called molecular fossils, because they presumably are showing the history, of the genome, of that individual or that species genome. Now, one particular class of, junk DNA sequences are endogenous retroviruses, and these are believed to be the genetic remnants of retroviruses that infected germ cells of an organism, where that retroviral DNA would be incorporated into the genome. At that point, it's called an endogenous retrovirus, or that process is called endogenization. And then the idea is that ERV would undergo mutations that would then render it non-functional, but yet it's retained in the genome. And the human genome has about 8% endogenous retroviruses. And so for somebody that holds to a design perspective, not only do you have to show that there's function, but you also have to show why these ERVs, if they indeed are functional, why they have similarity to viral genetic material, right?
So and if I've got this correctly, the claim is an ancestor, which was an ape, was infected by a virus of the retroviral class, which means that it inserts its own DNA into its DNA, and it happened to infect its germ cells, so its sperm or ovum, and then its offspring, hence, since this was integrated in its DNA, inherited it, and since we have it, we have to be offsprings of that ape. That's it. That's why it's specifically interesting.
Yes, yeah. And on the surface, it seems to be, frankly, a compelling argument. But what we've discovered is that endogenous retroviruses seem to have function, and in fact, there is just a flurry of papers that are now being published identifying functional significance to endogenous retroviruses. And it turns out that in a nutshell, they are functioning as an antiviral system. When ERV sequences get expressed, they trigger events that activate the innate immune system, already helping to ward off viral infections. They play roles. Actually, there's just a new discovery that ERVs actually play a role as anti-tumor agents. Sometimes tumor cells can evade the immune system. They
so
become
even without a viral infection, they sometimes just trigger and attack tumors.
Yes. Well, they don't attack tumors, but they cause the immune system to attack tumor cells that are invisible to the immune system. And what happens is there's this enzyme called P53 that normally suppresses expression of ERVs, but when the cell is under stress, will actually cause ERVs to be expressed. And when they're expressed and they start to produce essentially viral genetic material, it makes the cell seem as if it's just been invaded by a virus, which then invites the immune system to come and essentially attack it. And so it's actually an anti-tumor mechanism that helps to the immune system to detect tumor cells that otherwise would be invisible to it. And there's now researchers thinking that if they can target the P53 protein and get it to express ERVs, that this might be a very effective next generation cancer treatment, where you're using your body's own innate immune system to attack the tumor cells, but ERVs are factoring centrally into this mechanism.
So what you're saying is that if a cell knows that it's part of a tumor, but nobody's noticing, it says, hey, I'm infected with a virus through its ERVs, so that the immune system will come and kill it.
Yeah, so even though it's technically not infected by a virus, it gives the so this is called viral mimicry. So this is the name given to this phenomena. But this is just one example. You mentioned the koala, or maybe it was off air when we were chatting, the koala endogenous retroviruses. People have now discovered that these retroviruses actually will disrupt retroviral infections. So during the endogenization process, where retroviral DNA gets incorporated in the koala genome, these koala ERVs will actually express, and through a mechanism known as retrotransposition, insert and disrupt these retroviral sequences. So it's another means by which an antiretroviral process is initiated, there's also some endogenous retroviruses will actually produce the components for a retrovirus, but they're non-functional components, but they gum up the retroviral biology, its life cycle, preventing the retroviral particles from assembling. So there's a variety of different antiviral mechanisms that are being discovered that these endogenous retroviruses play. They seem to be activated at very high levels during the very early stages of embryonic development, when the embryo is in when there's only a handful of cells in the very early stages. And the thought is that this activation may be triggering genetic mechanisms that are critical for development, but also that it's giving an antiviral protection to the embryo, which is highly vulnerable at the 2, 4, 8, 16, 32 cell stage. In the very early stages of development, that embryo is highly vulnerable to viral infections. And so this is another way to ward off viruses infecting, the embryo at the very early stages, when it's, again, completely vulnerable and it lacks any kind of immune system.
Is it also true that one of the arguments for ERVs being part of evolution was that the some of the proteins, the retroviral proteins, are not needed for humans, and then a discovery is that actually some of those proteins are essential for placental development, because the embryo needs it to puncture through the walls of the womb, just like viruses just go and integrate themselves into cells. Is this also true? So that's also one of the reasons they are important for expression during embryonic development, maybe.
Yes. Yeah. Well, you're talking about the syncytins, which again are these fusogenic proteins that are critical. And what's interesting is evolutionary biologists argue that this protein was actually co-opted from an endogenous retroviral infection very early in mammalian evolutionary history, and led to the formation of placental mammals. But the argument is that
that is really provocative. So mammals are essentially sick animals.
Yes,
that's very pathetic,
Yeah. So but the idea is that this is such a critical step in development that you don't have the luxury of playing around with it, getting it to work. It's got to work and work perfectly at the very onset. So in and of itself, given how critical, this is to the reproductive success, to me, that in and of itself makes it very difficult to envision how evolution could explore, this possibility and evolve a highly optimal system that would exploit, a retroviral gene in this way. To me, I look at this as an example of modular design again, where the endogenous retrovirus is going to have fusogenic proteins. This placenta, this process of placental formation requires a fusogenic protein. So why not use comparable designs? And what we think is really homology, and evolutionary homology, is actually, again, common design. But what the kicker is that evolutionary biologists argue that this emergence of syncytin as this fusogenic protein in placental development must have happened independently multiple times, on the order of 10 or 11 different times, because
convergent evolution kind of thing. Yeah.
Yes, which again it's remarkable to think it happened once, to think it happened multiple times independently. So so this is a really good point that you bring up, because this is another example of where we're seeing function, where we shouldn't necessarily see function, right, from an evolutionary perspective.
I'll just add a small thing before we move to the closing here. Can we say, because I've got your anthropic thing now sorted out, can we say that, I'm being speculative here, that maybe It's because it's the same God who has designed viruses, designed bacteria, and designed animals and human beings. He's just using the same design. Maybe ERVs are not indigenization of viruses altogether, or maybe some of them is not. Maybe they are there because the one who has created the viruses also does not want viruses to destroy life. So he creates the virus and the antivirus, regardless we have vaccines or not, regardless we have cures or not.
Right.
So at least it will not eradicate life. To me, this looks like a best explanation. Does it look consistent with your anthropic principle thinking that common design can also be across creatures that are so far away, like a virus and a human being?
Yeah. To me, I think when you stack it all up, really the best explanation is ultimately design. Because when you look at the fact that the universe has a beginning, we see design in the universe. We see design at so many different levels and can be articulated in so many different ways. When we look at biological systems, we struggle to understand how chemical evolution could produce, the very first cell. We struggle to explain key transitions in life's history and the origin of novelty. When we put it all together, to me, the best explanation really does seem to be design. And what I see is many evolutionary biologists really isolating their discipline from every other discipline. And so in being under the influence of methodological naturalism, they look at shared features and they say, well, this is clearly prima facie evidence for common descent. But if you number one, look at the evidence more globally across the scientific disciplines, and at least allow for the possibility of design, I think you really are pushed in the direction that the overall best explanation, is design, where there's a creator that's intervening in some way, at key points in life's history.
Is this going to be undermined anyway by the matter that the same locus or comparable locus for where ERVs are in chimps and humans are the same, or this is not a significant issue?
Well, I don't think that is necessarily an issue, because it very well could be that the location of genes in the genome may have functional significance. Right now, I don't know that we can say that that's firmly established, but I don't think we can say that it's not been established either. And so this is really where these two different paradigms make very different predictions, where we would expect there to be, some kind of functional significance to the location of, different genetic elements, or there's at least an explanation for why there are these shared locations that don't necessarily have to involve a common descent.
It takes me to the next question, because I see it over and over again. And the reason I want to raise it with you, I hope you can be generous with 10 more minutes to get this. Yeah, is that I see it's very unfair to the lay people, and even to sophisticated people who are not specialists, that whenever a new line of evidence or discovery comes, evolutionary biology will just grab to it, build all those explanations on top of it, say it's very well supportive to their paradigm, and start shipping it in all sizes and forms and shapes. And some people are really shaken by this, and some have the patience to wait to see how it unfolds. Like you said, just ERVs have taken a completely different line of discovery in the last few years, and some are not. So is it fair, in your opinion, for evolutionary biology to rush into making such speculations to support evolution in fields that are still very young and not understood, like ERVs, like the significance of locations? And is it fair that they are doing this while they claim that 98% of the genome is junk just because they don't understand it? And then they're making inferences based on something that they say they don't understand. Is this fair? And why are they doing this, in your opinion?
Yeah, well, I don't think it's fair, and I think, for example, when we look at the science of genomics, we're absolutely in our infancy, and for anybody to make the assertion that we somehow understand genomes sufficiently to start making certain assertions that this is consistent with evolutionary theory is highly premature. In fact, the more and more that we're learning about genomes, even at this very early stage, the more sophisticated genomes seem to be. And I suspect that eventually, to really understand what's going on with gene expression at a genome-wide level, it's going to involve very sophisticated work in systems engineering. And in fact, I even suspect that what we're going to discover when we start looking at genomes from a systems engineering standpoint are principles that engineers will be completely unaware of, that are going to trigger development in systems engineering as people begin to dissect what's happening at the genomic level. But I think your point is spot on, is people are assuming evolution is true and they're interpreting everything from that lens. And unfortunately, I think what happens is that we're very quick from an evolutionary perspective to see something that doesn't make sense and to declare it to be a flawed design that's just a product of an unguided, historically contingent evolutionary history, and leave it alone. Whereas, and then what we discover later is that, wait, there's actually a reason this is that way. There's function, and this is stultified and delayed scientific insight. We've known about ERVs for decades and decades, and nobody studied them really, because the thought was this is junk that is the origin of an ancient viral infection, and if there was any studying of those, it was basically maybe to classify these systems, try to use them as a tool to discern evolutionary history. But it was really by accident that people discovered, not because of evolutionary theory, but in spite of evolutionary theory, function. And now we realize that our perspective was completely wrong. And now there's a flurry of papers of people, it's almost hard to keep up how many papers are coming out ascribing function to ERVs, and so this is just showing how, really, this evolutionary paradigm has stultified science, and a design framework would have right away said, even though we don't understand it, even though it doesn't make sense, we're going to press in, we're going to try to look for possible function, because we recognize that design is inevitable, and the question of, how is this system working? So I think the design approach is more fruitful in the long run for science.
Is it then a fair statement? It's something that I say. I use it to tease evolutionary biologists. Well, let me let me ask you if it's fair, according to what you've just said. Is it fair to say that the evolutionary paradigm pushes a scientist more to be lazy than to be inquisitive? Is this fair?
I wouldn't know. I don't know if I would say that it makes biologists or scientists lazy, because they're very hardworking people. But I think what it does is it forces you to.
I mean, it will tell him, maybe this is just junk. You don't need to look for an explanation, and evolutionary theory will give him leeway for that.
Yes.
But a teleological standpoint will not give him leeway for that. If there is something, you need to find the answer, because everything is for a purpose. This is what I mean. Yeah.
Yes. Well, actually, I can give you an example where that actually played out. Everybody, I think, is familiar with Craig Venter. He's somebody that I deeply admire and respect, though his worldview is very different than mine. But he was involved with the Human Genome Project in the very early days and was extremely frustrated with the lack of progress, because he felt like that most of the genome is junk. We shouldn't be wasting our time sequencing the genome. We should go after only those regions of the genome that are transcribed. And with his argument that if it's transcribed, it must be functional. And he published a paper very early on in the process where he looked at what are called express sequence tags, and he discovered, a huge number of genes that are expressed in neural tissue that nobody even knew existed. And so he actually left NIH in a huff, being very frustrated with the approach that they took, and formed the Craig Venter Institute. Well, Eventually, he becomes part of as a part of a private company, a competitor with the public Human Genome Project. But that was an example of where Venter took that very approach. Instead of doing the hard work of sequencing the genome and then going back and asking the question, what parts of this genome are functional, his approach was, let's just recognize it's junk and short circuit the process
and call it a day.
yeah, and call it a day. And so that was a real example of exactly that thing. So I think what happens is the evolutionary paradigm dissuades you from asking questions that really need to be asked.
It takes us to the exactly next thing. Where is this going to take us? What's the future of evolution versus creation, in your opinion? And let me just tell you all the four questions and have you answer them in one bunch. They're all the same thing. You're just hitting at it. Yeah. Is science taking us more towards creation or evolution, in your opinion? What is the future of this evolutionary paradigm, given what is happening? Finding out more and knowing that we had bad preconceptions about things and presuppositions. And are more scientists speaking out, in your opinion? And is this going to be a trend? And finally, given this, just like the example you've given now, what is your advice to students and even postgrads and even researchers, postdocs working in this field, in biology or in medicine? And in your opinion, would accepting evolution or creation be the thing that helps them produce better? Yeah. So let's, I'll give you the whole thing. It's a bit unfair, but I also don't want to take much more from your time. So let's have this in a nutshell. And
Sure.
Yeah.
So to me, I think, more and more, we're not going to be able to escape the idea that, biological systems are designed. And we're beginning to see work in synthetic biology, which is the area where engineering and biology are melded together, where the goal is to design intentionally non-natural organisms, primarily for technology purposes. That is going to reveal, I think, more and more evidence that biological systems are designed. As we try to design these systems, it's going to become more and more apparent that biological systems are designed. We're seeing an explosion of work happening in biomimetics and bioinspiration, where people are building new designs and developing new technologies inspired by biological systems. And again, this is going to, I think, really highlight the beauty and the elegance of design. I think we're going to see more and more scientists really being increasingly sympathetic to design, as a result of what's happening in these areas. I see more and more young researchers that are open to the idea of design. Unfortunately, many of them have to operate kind of in a clandestine way, where they can't really expose, their true interests.
Undercover.
Yes, yeah, because let's face it, again, biology is anti-teleological. Methodological naturalism is of a strong influence, and today, ID is viewed as such a threat, that many people in the scientific community are looking to root out people that are sympathetic to design. So, for a young person, you want to be very careful not to be labeled, too early in your career as sympathetic to design, and so you want to really, do your best work, and show that you have a mastery of evolutionary theory, and yet by the same token, I think, employ design principles in your research. And through that success, I think you're going to earn the credibility that will give you the opportunity, I think, to fully, disclose your perspective. But, nobody's ever been disappointed by viewing biological systems as designed when they pursue research effort. I've never seen anybody be disappointed by thinking about biological systems in a design framework. In fact, I've seen the opposite, where it's a very fruitful framework. and I think the reason for that is because these systems really are designed.
I think this was a great insight. I pray to God Almighty that this good omen is actually what takes hold, and that people would recognize the truth. And as in your experience, science takes them to faith rather than away from it, regardless of the pressures during the education, but to make the real benefit of education, which is to find God. Faz, I cannot be as thankful as I should be to this great interview. Sorry if it has taken a little bit more time than.
I've enjoyed it thoroughly.
I've enjoyed it more than anything, so I'm glad you've enjoyed it too. And I will have to ask you to be here again after your book is issued, or even before, because I cannot have enough of your great ideas and insights. Thank you very, very much for being here, and I hope this interview will be beneficial to so many people who want to learn about science, but most importantly, how science can take you to faith.
Well, thanks for having me, and we're kindred spirits. I can already sense that, and so I'm glad to get to know you, and I'm happy to do anything that I can to contribute to your program and to your YouTube channel. So it seems to me to be very worthwhile. So
thank you very much.
Much success to what you're doing.
I'll hold you to that offer, and thank you very much, Fuz. Everybody, this was Dr. Fuz Rana, one of the great minds in science and faith. Talk to you soon. Please also visit his website and give a good like and share to this video, and share it with your loved ones, and let's spread this to the ends of the earth. So thank you very much, and peace be upon you all. And we are going offline. Just let me put the extra there so we can put some links. And yeah, here we go.